Thank you, Rupert, Sarah, and to the committee for inviting me today. What I thought is to provide an overview of glutamatergic modulators and work on biomarkers more on a macro level to set the stage for the other speakers, which will get specific into other glutamatergic agents. And for Lisa, I think she's around somewhere, that will talk more about the cellular molecular mechanisms. Okay, this is my funding. I work at the intramural program at NIMH. So the objectives of today is to become familiar with translational research on treatment-resistant depression, specifically with rapid-acting agents, RAD, to understand the neurobiology of promising therapeutics for treatment-resistant depression, and to understand the pharmacology of promising therapeutics that have a rapid-acting onset of action. There is a considerable need to develop novel therapeutics for TRD and for suicide. We have the adverse effects of depression, including disruption to personal, social, occupational life, and there's a considerable risk for suicidal behavior. We also have many treatments, including psychosocial, pharmacological, and neurostimulation. My talk will focus mainly on the pharmacological and specifically in glutamatergic agents that have promise for rapid-acting onset. We know that there are many limitations with our current antidepressants, including very low remission rates and a considerable lag of onset of therapeutic effects of several weeks, if not longer, for both depression and for suicide. This figure depicts a major depressive episode. The natural course is about 6 to 9 months without treatment. What happens with treatment is that you shift the curve of response towards the left. So when you start treatment with a monoaminergic-based antidepressant, you have an onset of action of about 10 to 14 weeks, which is helpful, but you have the risk of disruption to your life and risk of suicide. So next generation treatments will have an onset within hours or a few days at most, and that is very feasible with the prototype ketamine and how to deliver safer alternative. But ketamine does have its limitations. We can learn from it, use it as a tool for biomarkers, but we need to understand the cellular molecular underpinnings of these effective rapid-acting agents so we can develop better treatments. Now, this is like as Rupert mentioned, when we showed up at NIMH in the 2000, Dennis, Charlie, and Husseini Manji, this was an early figure, quite simple, and this is the original published in 2002. And you can see here the tripartite system. You have the presynaptic neuron, the postsynaptic neuron. You have glia. You have glutamate that kind of comes in back and forth, and a few candidate drugs back then. We were very excited about memantine. We were very excited about riluzole, and somewhat excited about ketamine, but there was concern. And I'll bring you up to where we are now with treatment. Now, the original, one of the original studies besides memantine, riluzole was with ketamine. Not going to go into detail because you have many wonderful lectures that are going to go much into detail. The main point here is that it has a very short half-life with a single administration, and it binds to the PCP site. And I don't know if you can even see here. Can you see here? I don't think this is working. Maybe. Oh, there we are. And within the transmembrane domain, you see the site where ketamine or S-ketamine here, I can't see from far away, binds. Here's the cartoon that's within the PCP site. And during the administration of ketamine, 40 minutes, or depending on the duration, you have impairment of learning, plasticity, and memory. That's all I'm going to say, but I'm going to go into more. This was a study in treatment-resistant depression, the mid-year 2000. Here, the question the question was, if you block an NMDA receptor with an inhibitor, do you bring about a rapid antidepressant effect? And the answer is yes. Because of ethical reasons back then, we had to study people who had failed 6 to 7 antidepressants, and about half of the patients had previous suicide attempts. So, and you can see here, depression scores, higher number, greater severity of depression, time in minutes or duration. This was unheard of in a sense that most of the studies are by weeks, and here we're doing assessments in minutes. And you can see a rapid onset within a couple of hours, lasting a week. Towards the right are the response rates You see with our monoaminergic-based antidepressants, as I mentioned, 65% taking a pill every day for 6 to 8 weeks in non-treatment resistant depression. Here you can see rapid responses within a couple of hours. And for most of you who already do work with ketamine, you know about this. What I want to point out is that the dissociative side effects are very short-lived, 40 minutes after the infusion or so, and that the half-life is only a couple of hours. So why were we having this ongoing, sustained antidepressant effect lasting for 1 week, 2 weeks in some individuals? And that's kind of what I want to focus on the rest of my talk. Now, what are the pharmacological classes of agents with rapid onset? And they're different in play now. But there are 4 major groups: the glutamatergic modulators. The prototype is ketamine, but besides being an NMDA receptor antagonist, it also has effects on mu and kappa opioid receptors in other systems. We have the neurosteroids, brexanolone or Sage 547, GABA, NAM, or negative allosteric modulator, and also has effects on other receptors. Then we have the group of the opioids, which is becoming more interest, AUX 5461, and others like Rimalda compound. And then we have the serotonergic hallucinogens. The prototype is psilocybin 5-HT2A agonist. Now, this is a very preliminary, but you start thinking, okay, can we learn from these agents to try to hone down on what might be relevant to rapid acting agents? They are different drugs. You can see that they initiate at different places and receptor opioid, of course, opioid drugs on opioid receptors, glutamate on NMDA, AMP, and so on and so forth. But they start converging in the plasticity cascades, and more downstream, we see that they have effects on network reconfiguration, including increase in neurotrophins. We have protein transcription, neurogenesis, cytoskeletal reconfiguration. Now, this is all theoretical. Some of the studies are very early, but you could see that they tend to overlap in the intracellular plasticity cascades, which lead eventually to this network reconfiguration. Now, I am not suggesting that there is one mechanism, but different potential possibilities that they overlap on. Now, focusing more on the macro, I'm not going to get into the cellular molecular, well, Lisa will do a great job on that, but just to give you an overview of some of the different theories, we have one that ketamine has effects on GABAergic interneurons, on NMDA receptors, decreases GABA, leads to a hyperpolarization or excitation of pyramidal cells, the so-called glutamate burst, AMP activation, and then a series of downstream signaling cascade changes, induction of neuroplasticity, spine growth, neurite growth, synaptogenesis. Other theory, which is more direct effects by blocking NMDA receptor, eukaryotic elongation factor 2, I'm not going to go into that, induction neuroplasticity, spine growth. You can see where they start converging, at least some of the theories. Then, in order to avoid the NMDA receptor, you can go more downstream, for example, inhibit mGluR2-3 signaling. You produce this glutamate burst, and you perhaps avoid some of the dissociative side effects or abuse possibilities. The other candidate drug in play is 2R6-RHNK, the son of ketamine or hydroxynorketamine, also leads this glutamate burst, the same downstream mechanisms I mentioned. The serotonergic psychedelics, you have a downstream GPCR pathway, it's involved, and you might have increases in glutamate release in the same mechanism. And then you have a possibility, brexanolone, we don't know for sure. It's a negative allosteric modulator, but may involve some of these pathways. So this is very broad overview. Now, what I'm interested in, and I'm going to get a little bit, is this this interplay between inhibition and excitation produces EEG or MEG changes, and you can measure this. And so it could be what we refer as the gamma power in the 30 to 50 hertz range. Could be a potential marker that you can use in rodents and in humans to help in drug development. So this is where we're in the 2000s. This is how the system is right now. This is a good problem. Jerry did a very nice talk last year. It's online and you can go into that. What I'm going to talk is more on the bigger picture. What are some of the candidate drugs? If you want this paper, I can send it to you. So we already talked about GABAergic interneurons. You have the mGluR2-3. We talked about signaling cascade pathways. Racemic ketamine, I'm going to keep on referring. There's a difference between racemic, S-ketamine, and R-ketamine. They are different drugs in some ways, and they have similarities in other ways. Here you could see that one possibility I'm going to get into a little bit later is liver metabolism. Then the son of ketamine, or 2R6R-HNK, has AMP activation. There's some drugs such as mGluR25 antagonists co-localized within MDA receptors. And so that's another way of perhaps tapping into the system. You can look at TAK-653 as a more AMP activator, direct effects on that. Extrasynaptic MDA receptors, another possibility. Some believe that racemic ketamine is involved in that. We have NV5138, and mTOR activator. I think Chadi is going to be talking maybe about this a little bit later. And I'm trying to introduce the speakers as they go. And then you have broad effects on an NMDA receptor. You have a long list of drugs here. Dextromethorphan, you have nitrous oxide, a very nice recent study that was published showing comparable efficacy of 25% and 50% in fairly rapid onset antidepressant effects. You have MIJ821 I didn't have space to put it here, so I put it in this box here. You can see how complex. The glycine site is a way of fine-tuning the NMDA receptor complex in theory so that you could have the therapeutic effects without the side effects. And here we have a few, D-cycloserine, repastinel, apomestinel, and then 4-chlorokynurene, some with mixed results. And of course, the NR2B receptor. Now, I'm interested in mechanism in addition to potential side effects or complications. And here we have that ketamines, it was proposed that opioid signaling is involved in ketamine's effect. And so this is the paper by Nolan Williams. And you can see here, subjects were pre-treated with either placebo, say a placebo condition. And you can see here that it did not block the antidepressant effects of ketamine or naltrexone, where you see that there's a attenuated antidepressant effect still there, but not as much as when you pre-treat with placebo. And they suggested that probably the opioid receptor is important to therapeutic effects, but you don't see the blocking of the dissociative side effects. And so that, raised some concern, you're going to use this clinically and so on and so forth. A subsequent study by Matt Klein suggested, well, it's not direct effects on mu-opioid receptors, but it's NMDA and opioid signaling. And perhaps those two are important to rapid acting agents. And so I think that's going to be important in drug development, that it might be possible to find compounds that modulate opioid signaling, but without the abuse liabilities. I'm introducing this as a better understanding that we have the enantiomers of ketamine. We have racemic ketamine. And keep in mind, when you give racemic ketamine, you're given 24 drugs. It has 24 metabolites. So, it might be that it's not just one drug, but it's the combination of drugs that makes it so unique, kind of like ECT. It does a lot of different things. We don't know yet. Breaks down to racemic norketamine, dehydronorketamine, and then there are many different metabolites. The summary here is pay attention to R-ketamine is metabolized to 2R6R-HNK, and then S-ketamine to 2S6S-HNK. In preclinical studies, both of these have antidepressant effects. Some of us have decided to go the R pathways, others are pursuing the S pathway. I'm not saying one is better than the other, but they may have roles in different conditions. So we're looking at mechanism, but in addition, we have to address the issue of potential abuse liability, as been reported in the Williams paper. And so tolerance develops, or tachyphylaxis to the anesthetic and pain effects. But when it's used recreationally, it's usually several grams a day. As you know, as clinicians, we give much less. We give 30 to 50 milligrams a couple of times a week for a short period of time, and then we spread it out. But nevertheless, there has been use of ketamine. About 2 to 3 million people in 2006 was reported. And in some countries, it is abused. But of course, there's a REMS system where you can monitor its use and supervise it, and that addresses many of the concerns. But nevertheless, we were interested in trying to find the magic target of ketamine. and probably there is none, but here you can see that this is a study looking at 6,000 to 20,000 different targets. This is done in Mike Michaelides' lab. Jordi Bonaventura was the lead author. And what we looked here at was that S-ketamine, R-ketamine, looking at either 100 nanomolar and 10 micromolars. You can see here all these little letters and numbers are the different targets, enzymes, so on and so forth. And what you find is, not surprising, PCP site I already mentioned. Both enantiomers bind to PCP site. But what we find is that the S enantiomer seemed to have greater effects on mu-opioid receptor than the R enantiomer. So that was quite intriguing. Then you use radio-binding assays. This is the ability to displace something that's potent for that target. Here NMDA is MK801, and in orange, might be hard to see, is S-ketamine is more potent an NMDA receptor than is R-ketamine. But if you look at opioid receptor, and I've summarized, I summarized here in the bottom, here that you have greater affinity for mu-opioid receptor for S compared to R, and kappa receptors for R compared to S. So as a right, sigma receptors have been implicated in depression as an antidepressant. And so what you find here is that R is more potent than S at these targets. The next is, and this is just a summary of the paper, but we looked at FDG for the deoxyglucose, PET imaging here in rodents. And then the top is S-ketamine, bottom is R-ketamine. With S-ketamine, you see greater activity in medial prefrontal cortex where the opioid receptor resides. With the R-ketamine, you see actually decreased activity in the paraventricular and habenular region, which is interesting because there's, as you know, increased activity in habenula linked with depression, and with EBS, deep brain stimulation can knock it down, and that's the anti-reward system. So potentially, R-ketamine might be helpful in anhedonia, for example. This is out of radiography study in rodents, and this is the prefrontal cortex, and you can see here in S-ketamine at baseline, and when you pre-treat with naloxone, which is a mu-opioid antagonist, you diminish it significantly, suggesting that mu-opioids are involved with S-ketamine, but you don't see much change with R-ketamine. It's not blocked. So here is the figure, the histograms. We see it increases with morphine, and it's blocked. S-ketamine is blocked with naloxone, but no significant changes on R. So next is you could conduct a series of behavioral effects of the enantiomers, looking at locomotor activity, condition place preference, and self-administration. And this has been used for opioid drugs and psychostimulants. And here, just focus on orange is the S-ketamine, and then in red, I believe, is R-ketamine. You can see with S compared to R, greater acute locomotion, sensitization, and cross-sensitization. Condition place preference on the bottom. You see it's greater for S-ketamine, no significant changes for R. And then the lever press is when you have a greater desire to have the drug. Here it's greater with S compared to R, no significant changes. But what's interesting with these drugs, in contrast to opioid drugs and psychostimulants, is both with R and the S enantiomers, there's extinction. So there's not a greater, and probably Celia, I don't know what she is, but she will go more into detail about this. There's not this need to keep one person. So there's something really unique and different about these drugs compared to the traditional opioids and psychostimulant drugs. So to summarize, the intravenous drug self-administration models, rat self-administered S but not R. The S but not the R is associated with changes in classical behavioral paradigms associated with opioids and psychostimulant drugs. But there seems to be this, that mu-opioid receptors and kappa were affected more by the S than the R. And there seems to be this, this divergence in terms of the properties of the behavioral pharmacological properties of both S and R-ketamine, enantiomers. So what we're saying is that they might be different in some ways, similar in other ways. Okay, now the next part of the talk, I think I have 15 minutes or so, is that we're trying to understand more the mechanism of ketamine. And so I'm focusing more on the macro part And here, what we've developed is a multi-scale systems biology approach where we integrate a wide range of clinical, behavioral, and other techniques to understand better how ketamine works with a single administration and repeat administration using different technologies before, during, and after the administration of ketamine. Here are different modalities we used. But first, I want to summarize in this cartoon some important concepts. So we talked one possibility of how ketamine might work, blocks GABAergic interneurons on NMDA receptor and GABAergic interneurons, leads to this glutamate burst. There's evidence for ketamine of increases in gamma power when this happens, but not for scopolamine. And you have an increase in spines, as we mentioned. But there are other drugs. We can stay away from the NMDA receptor, hopefully not have the abuse properties. You can look at mGlu2 antagonist. You can look at the HNK, the son of ketamine, also does this glutamate burst. But what these different compounds have in common, some of them is AMPA activation. So why is this important? Well, in a study done in Husseini Manji's lab, some time back, this is immobility. The forced swim test decreases in immobility, suggests antidepressant-like properties. You see that with ketamine compared to saline. But if you pre-treat with an AMPA antagonist here, you block the antidepressant effects of ketamine. And so that seems to be a property of some of these other compounds. In addition, you have this excitation inhibition seems to be represented or a phenomena could be gamma power, this excitation inhibition. And I'm going to talk a little bit about that. Now, there are ways to examine, or tools to examine this plasticity, synaptic potentiation. There are EEG, there are slow wave sleep, and TMS, and we're doing that in a recent study, in a study that's going on now. We call that the new Barrick study. Since I work in government, we don't have enough money to pay people to come up with clever acronyms. So this is the only thing, and you can guess why we called it New Barrick, because it was a particular time in the United States, where it's quite complex, but we are doing MEG in the subject while they're getting ketamine. We do fMRI and EEG at the same time they're getting ketamine, and then the same subject goes over and crosses condition. And then they get repeat administration, low dose or high dose ketamine with biomarkers, what I just mentioned. Now I'll present some of the results today. This is just evidence at the circuit level. I'm going down and then hand it off to Lisa, where this is double-blind, single administration, unmedicated subjects. We obtain 3T MRI at baseline. We give ketamine, and then 2 days later, and then 10 days later. What we're trying to capture is the onset of the antidepressant effects and the offset. And you can see here, specifically looking at the triple network, default mode network, central executive network, and the salience. So it's believed that the default mode network might be increased in depression and decreased central executive network, or disturbances with salience network, the ability to switch back and forth between these different systems. You find that baseline, our patients are depressed, moderately severely depressed, a score of over 30. And with treatment, of course, scores go down. And then the effect of ketamine single administration wears off. Everybody knows that curve, right? So what we wanted to do is match the symptoms with biology. And here you can see at baseline, this represents the difference between patients and healthy control subjects. And you see greater activation of Insula, salience network. At the peak of antidepressant effects of ketamine, 1 to 2 days, you see there's no longer a difference, suggesting normalization. The effect of ketamine wears off. You have a return in the activation of Insula. So a very nice way of mapping on-off effects of ketamine. In green, no significant changes in saline. The next is work on anhedonia. So there will be some presentations here. Earlier studies suggest rapid anti-anhedonic effects of ketamine, sustained, involved in pleasure. It's a cardinal symptom of pleasure. And what was interesting, and I hit here, who did the study was interested, was the cortical striatal circuitry. And so these were 33 unmedicated subjects with depression, TRD, and then 25 healthy controls. And what you look here are the aspects of anhedonia: emotional, cognitive, motoric processes, and these are the 4 seed regions. We have dorsal caudate here, ventral stratum, ventral rostral putamen, and the dorsal caudal putamen. Here, what we looked at are the global connectivity changes. And this is, you can see the four seed regions, A, B, C, D. If we take one example, ventral stratum with dorsolateral prefrontal cortex, and you look at connectivity, you have green placebo and orange ketamine. And generally what you find is when you give ketamine to healthy controlled subjects, they have a decreased connectivity. But when you give it to patients with the treatment-resistant depression, they have increases in treatment, in connectivity, I'm sorry. And so those are the opposite effects you see in healthy controlled subjects and in individual TRD. Here, towards the bottom, you look at the correlation, the relationship between the changes in ventral stratum connectivity, dorsolateral prefrontal cortex, and improvement in depressive symptoms. And so several of these nodes, and including anhedonia, are in the direction you expect with the reward circuit. So that's another intriguing finding. Now, next we go down a little bit, get them off closer to the cellular molecular, is at the circuit level. Here, what we looked at is MEG, EEG, gamma power in the 30 to 50 hertz range. So we get at baseline before ketamine, 6 to 8 hours after the, when the antidepressant effects take off and you no longer have dissociative side effects. What is, as I mentioned, the gamma power is within the 30 to 50 hertz range, maybe higher, but you could see here these changes in early visual cortex in the frequency of 30 to 50 hertz range. You can find gamma oscillations in visual cortex, auditory cortex, which is nice because then you can study these different systems. And why are gamma rhythms important? Well, they're correlated with action potential generation, involved in sensory perception, cognitive processes, information encoding for memory, and hippocampus. And so many important processes. And we are trying to find objective measures beyond the MADRS and the Hamilton scores. And that's the point of this. Now, what's interesting is where are these generated, gamma? Gamma is, we don't know exactly where, but there are these micro circuits one is called the interneuron network gamma. You can see here are inhibitory neurons, the excitatory pyramidal cells, and you have this inhibition excitation change that I already talked about. When you have pyramidal cells with the negative feedback, you have the ping pyramidal interneuron network gamma. So these are microcircuits. Oops, sorry. At resting state, going back to the triple network, when you give ketamine, you find on the top, depressed subjects, bottom healthy control, you see increases in gamma power with all these drugs, but this is 6 to 8 hours after giving ketamine. This suggests non-NMDA or maybe AMPA, activator cortical excitability. Here, towards the right, you see increases both in depressed subjects with ketamine and healthy controlled subjects, but with the subjects treated with ketamine, depressed subjects, you see it approaches the baseline of healthy controlled subjects treated with placebo, suggests normalization. So those increases in gamma power potentially could be a very important biomarker. Where are, in areas of the default mode network, Insula, for example. What we find here, and this is not ready for prime time or clinical use, this is more at the lab level, but you find that the baseline gamma power before they receive ketamine moderates the antidepressant effects. If you have lower gamma power, you're probably going to have a better response. If you have higher gamma power, you're not going to respond or actually might get worse. That's the operating theory. Now, you can induce plasticity or measure plasticity, in this case, using, if you recall, gamma power sensory cortex, right? You do a pneumatic device, in the index finger, and you produce changes in sensory cortex. So there's active plasticity here. And this is referred to as stimulus-induced gamma power changes. We select the gamma power, the 30 to 50 hertz range. And this happens in anybody, you can do that simple task. And if you look at in this open label studies, no difference between non-responders and responders. But after ketamine, you see in responders significant increase in gamma power, stimulus-induced gamma power, suggesting plasticity mechanisms at play. Now, we then went and extended these results in, replicated these results in a control study And you find in responders to ketamine, in increase in stimulus-induced gamma power using the same task. In non-responders, no increases in gamma power, and in healthy controls, there's no changes. So this starts suggesting that maybe plasticity is important. Here you look at the correlation between the peak gamma ketamine placebo difference and the peak of antidepressant effects of ketamine, which is day 1, 24 hours. And you see a very nice correlation here. So again, these are early data suggesting that perhaps stimulus-induced gamma power changes plasticity is important. Now, moving on, we looked at evoked related potentials here, looking at 18 subjects with depression, 18 healthy controlled subjects. And you can see here, bait on the top is a control subject, and the pattern is different from a patient, for example, right? What we're here is trying to understand the antidepressant effects, who are going to be the responders. And here we use dynamic causal modeling for this excitation inhibition model. And when you use DCM, dynamic causal modeling, you estimate and make inferences about the regions that are important in depression. And what you do is you perturb the system, for example, a sensory task, as I mentioned, and then you measure that the change of that perturbation. And that's what we refer to as effective connectivity. You measure those changes and give you a sense on how might people respond. Going back to this microcircuits, the ping model, right? We're going to get information at the receptors of GABA and pyramidal cells. And you can create biophysical models where you have an indirect measure of NMDA, AMPA, and GABA receptors. And you can do these measurements and come up with these parameters on regional dynamics, because we sometimes take snapshots, but really these things are really rapidly changing. And when you do that, you can come up and then you can plot this in a trace determinant plane, and then have a sense on how these changes occur in individuals with depression. So for example, here towards the bottom right, you do these excitation to excitation, get a parameter and estimate, inhibition to excitation, and then you plug it in. And when you do so, you find that there's southwest direction, this trace determinant plane is associated with better response. And here you could see that it gives you an example. It's not too much excitation in the subject, too much inhibition, but it's really this interplay going on. And this is what's estimated or believed to call what happens at a regional dynamic level. And that will give us a better sense of who are those more likely to respond to ketamine. So we're now looking at a larger data set to see if this replicates. So we talked about plasticity changes at the 6 to 8 hour level, right? After the side effects of ketamine dissipate and when you're having acute therapeutic effects. So the question is, how early do these plasticity changes occur? And so we have looked at the Ubaric study and to get some preliminary data. So we use the same tactic. This is a different cohort. We just got some preliminary data where you do the air puff, you do activation of the sensory cortex. And what happens in the first 15 minutes, you get significant gamma changes already with ketamine, which is quite intriguing. Very early on. Now, what we're trying to do is, is it higher at 15 minutes and goes down, or does it come and go? Why is this important? Because some people argue that you have these rapid changes that occur, and then maybe it goes off, and then the system tries to compensate, and it's rapidly. We don't know. And so I think it's all about dose and timing, and to keep those in markers. Now, I think I have time for a few minutes and some recent data on the slow wave activity. So when you learn a new task, there are changes in plasticity genes in rodents. And here, a paper by Tononi and Sorelli, when you do this computerized task, you learn a new visual task, you have increases in slow wave activity. That is precisely in the same place as the motor cortex we were learning. So that suggests changes in plasticity. And so we reasoned here that if you have changes in plasticity, meaning insertion of AMPA receptors, you have antidepressant effects. And this is a bit simple cartoon. If you don't have insertion, you don't have potentiation or plasticity. And so what do we know about slow wave activity? When you give ketamine injections, it's associated with increases in synaptic strength, you have increases in slow wave sleep. So it could be a plasticity marker or potentiation. And so in a very early study we did, you find this is sleep. You do polysomnography, and what you measure here is delta in the 0 to 4 hertz range. And what you find here is the first cycle increases in slow wave activity with ketamine compared to baseline, suggesting plasticity effects going on. And early, we found that in the responders, it seems to be greater increases in those who respond compared to non-responders. So what I'm talking here is slow wave activity, gamma power, several different plasticity measures. And then very recently, we just completed this study where you look at differences when what you see in healthy volunteers is in non-REM sleep, a decrease in slow wave activity. You can see this very nice decrease in healthy volunteers, but the process is disrupted in patients with depression. And so the issue would be to normalize that. So does it normalize with ketamine and is associated with response? And here you look at on the left, this is a baseline of treatment-resistant depressed subjects in red versus healthy controlled subjects. I just showed you this. And you could see here that there is a significant difference. But when you give ketamine in this cohort, there is a normalization. So there's no longer a significant difference. So it suggests that slow wave activity is a putative marker of plasticity. And when you look specifically at responders here, you see increases in slow wave activity in responders. In the non-responders at 230 minutes, that's the early time point of antidepressant effects, you don't see a change here. So suggesting that there aren't mechanisms. And then finally, I'm going to stop with the last slide to show you, or 2 slides at a time, would be that we talked about this on-off effect of ketamine. And so we suggest that we, in Tal Gould's lab at the University of Maryland and others, Craig Thomas, Ruin Modell, and Pat Morris, what we identified were metabolites that last 3 to 7 days in blood after you give ketamine. So we wondered whether ketamine is actually a prodrug. We isolated 2R6-RHNK as a candidate drug, and in this cartoon, which is the last slide I promised, we give ketamine to rodents, we mentioned, and what happens in 10 minutes, you have 24 drugs or metabolites. And we reason that some of them are involved with side effects, addiction, and the others in the therapeutic effects. So when you strengthen carbon-6 bond, you produce diddeuterium ketamine. So it's another that's pharmacologically similar to racemic ketamine. You abolish metabolism significantly. You did inject it in rodents. You still have the side effects in addictive properties, but you don't have the hallmark, which is the sustained antidepressant effects in rodents. And so it does suggest that probably maybe something else going on. You isolate 2R6-RHNK, and you find at therapeutic concentrations, antidepressant-like properties in rodents, doesn't have NMDA receptor inhibition effects at the therapeutic concentrations, and no abuse potential. So we are developing this drug. So that's as we separate the wheat from the chaff, and that's a drug we're testing. So finally, I'd like to stop there, and thank you for your attention. I think I may have gone over a little bit, right?
Great.
We don't have time.
Yeah, no. So I think
Right.
We're going to have to.
Do you want me to unplug this?
If you stay put, and we'll take questions from the audience here and also questions online. So, does anybody have anything they'd like to ask? Celia.
Really fascinating to look.
So if I can just say, when you're asking a question, be aware that the seating mics work well. Speak nice and clearly, and speak towards the front. Sorry, Celia.
No. It's probably bad first person. No, I was just wondering, and yeah, thanks for a wonderful talk.
You say your name and where you're from? Oh, yeah, Celia. Yeah. Oh, yeah.
Yeah, so I was just.
I didn't recognize you without the nose.
Yeah. No, I was wondering if there was any differences if you looked with RNS and with any differences in things like slow wave activity or gamma.
No, I'm hoping to get our ketamine for my colleagues and friends here in the audience, we'd be delighted to look at that, because I think that's how we're going to learn. You know, about the abuse potential is one thing, but I'm interested in mechanism. Sure. Yeah.
Really interesting.
Thank you.
I'll ask a question. Most of the work that you've been doing is looking at fairly short time frames and doesn't look at repeated use of ketamine. Can you say what you might expect to see if you, with say, the gamma power, if you used ketamine repeatedly?
Yeah, so that's the new Barrick study. So we have, I'm sorry, I'm going to repeat the question.
Sorry.
So what biomarker changes do you expect to see with repeat administration of ketamine? We don't know. You know, it's one thing where if you gave ketamine daily, which we're not going to do because that's probably incompatible with the brain, maybe, who knows, but you have this on-off pulse treatment, you know, does the system reset? And then you're always doing this glutamate burst and gamma power, if you believe in that, you know, on over. We just don't know. There's some early data that people look at, you know, we'll see. I suspect that, you know, that we'll find some of the things early on in terms of gamma power changes with each infusion. It's probably not going to last long and then probably compensate. But I guess the question would be also, if you achieve remission, right, you know, is it wise to be giving ketamine to people who are remitted, you know, inducing these changes over and over again, you know? So that's something that we have to think about.
And then relapse.
Well, if you're euthymic in a remission, should you keep on giving drug, and what would happen? It's probably not a good thing. We don't know. But what would happen to the biomarkers with the system, you know, reset? Who knows?
Laurie.
Laurie Calabrese from Connecticut. Are there AMPA antagonists that we are unwittingly administering to our patients that are preventing some of them who don't respond to ketamine from responding in?
So the question, are we, if you recall, present some preclinical data, there's where if you give an AMPA antagonist, NBQX, you block the antidepressant effects of ketamine, suggesting that AMPA throughput is important. And there's a couple of studies, we're doing one, and then Yale is doing one, where you pre-treat with perampanels, an AMPA antagonist, to see if you can diminish or block. It's kind of like Chade will be talking about his rapamycin experiment with mTOR. So are there compounds or drugs out there? For example, I think topiramate has some, is that correct? AMPA antagonist properties. You know, would that diminish the antidepressant effects? There was already a study, if I recall, with topiramate, but it was done for cognition, I believe. And it seemed to have some effects, but, you know, but that doesn't mean it wouldn't necessarily block. We don't know. So yes, maybe somebody knows the answer. Yes,
Over here, and then this, you first.
Do you?
Say your name, please.
Dave Barwick, University of British Columbia.
You speak up.
Dave Barwick, University of British Columbia. Have any of these studies, or are there any others that you know of that have examined these effects in patients with coexisting severe substance use disorders, whether it's alcohol or stimulant use disorder? And what would you postulate that the potential benefit might be in someone that has a sort of a coexisting, contributing severe substance use disorder?
Yeah, so people with depression and substance use disorders, I want to steal the thunder from Celia. She probably will talk about all that. Krupetsky studies, right? You'll probably talk about it. And then her side, she has an American Journal Psychiatry paper just a few months ago. So she'll talk much more about that. But yeah, it's interesting. I would say, yeah, I'm gonna leave it up to her. Yes. Oh, sorry.
My name is Antonio Metastasio, and if we assume that ketamine is a prodrug, are you aware of any different pharmacodynamic patterns between responders and non-responders to ketamine?
So everybody heard the question, I believe. I don't know if online they heard the question. So I guess, are there could pharmacogenomics predict who might respond or not to ketamine? I think that was your question.
It is a prodrug.
Yeah, I mean, if it's a prodrug. So I think we did a study in 2012 looking at pharmacogenomics, very small. And we didn't find much, but we did find that when you combine with, if I recall, Depakote, that you have in bipolar depression, because it was bipolar depressed subjects, you probably have a diminished antidepressant effect from ketamine. Now, to my knowledge, I don't think there have been larger studies, maybe you know, okay, Sarah knows, I don't know, larger studies looking at that. I would say we have been looking at that. What we're doing now is doing some work in microspheres and looking to see if we can kind of identify. There is, to my knowledge, not any support for that. It's just too many different pathways. We've been trying to figure out how to study metabolism, and it's just really complex, like a mechanism.
We've got last question online.
Yeah, we have a question from Tiago Gil. Does the findings suggest a possible EEG during the infusion to modulate the dose and time and optimize antidepressant activity on the go?
Everybody heard that? Yeah. That's again, what I'm trying to stress is that we're interested, a lot of this is not for clinical use, and you go and purchase an EEG or an MEG, which are very expensive equipments, and then start doing your patients. I think this is kind of very early, I want to stress that, work to try to figure out mechanism. I would say there have been studies looking at MRS and glutamate, different doses, right? And you get changes early on, GLX, for example. It would be a nice study to see if you have differences in a dose finding study with EEG and gamma power, but it's not ready for prime time yet. Especially if you get a rapid response, you know, in a couple of hours, you're going to get better, unless you're going to get repeat, you know, for 2 weeks administration. So.
Good. Well, thank you very much indeed, Carlos. That's great.